CAR-infused group without total body irradiation, just to account for a control for all of our different variables that we have in our groups. And in this first experiment, we decided to run 2 different CAR T cells, our FLT3 ligand CAR, as I mentioned before, as well as our IL-12 CAR, to keep the groups small and our n's low for this very large and labor-intensive experiment. I decided just to remove total body irradiation itself as an entire variable, just so we see what if these CARs can actually kill without radiation. So, lo and behold, they do. So that was really exciting for us. So, in a sense, we performed the exact same experiments as I showed you before, and our IL-12 CAR T cells, V3 positive CAR T cells, eradicate these tumors entirely without any sort of hiccup, really. This table is not fully updated, but our FLT3 ligand mice do not currently see long-term survival. Those have dropped off to about, I would say, closer to 10% right now. So far, we still have a couple that are exhibiting some sort of long-term survival. But we've seen that essentially, by introducing this IL-12 local-regionally to these mice rather than systemically, we've been able to eradicate their tumor without any sort of lymphodepletion in these mice. This is very exciting for us because in many cases we haven't seen a lot of people looking at solid tumors in terms of CAR T cells in terms of solid tumor context. Can't remember his first name, but Brenchens, his group looks at armored CAR T cells in terms of hematological tumors as well as ovarian cancer models, and they've shown that in their ovarian cancer model, they've developed an IL-12 CAR as well, and they've showed long-term survival. So, in cases of GBM, this was very exciting for us. The other candidate experiments are currently in progress, so I'm not showing you anything just yet. But what I can say for now is that the CCL3 itself does not work, and the GM-CSF and CD48 ligand are still in progress. So it'll be exciting to see what those results yield in terms of no TBI. Yes.
So in this experiment here, the results you're showing, what happens if you just have the tumor and then IL-12 protein or tumor plus FLT3 ligand protein without the CAR?
So we haven't looked at the FLT3 ligand, so that's definitely something that we do need to look at. In some previous papers from Rosenberg's group, by delivering just IL-12, or let's say not a CAR T cell, just activated T cells, IL-12, and radiation, that actually helps, and that will kill the tumor. If you remove the variable of radiation and deliver only activated T cells and IL-12, the mouse dies. So in a sense, we're seeing that potentially the role that the CAR here is playing is providing some sort of targeting mechanism, allowing then our T cells to actually pick up antigen and then have some sort of way of either with that or being able, maybe there is some exhibited endogenous immunity of these T cells migrating into maybe the lymphoid system, lymph nodes, deep cervical lymph nodes, and then coming back and being able to kill the tumor. So we really don't know that answer just yet. But from what we've seen, IL-12 itself without radiation does not work. Yes.
So given the prognosis of these gliomas, you might argue it doesn't matter that a lot of these cytokines are implicated in CNS autoimmunity. So,
right.
I mean, if you look at the mice that survived, do you see autoimmunity or demyelination in these mice?
So currently from this current experiment, which is actually still running, we don't see any characteristics that has currently exhibited some sort of autoimmunity, but it's definitely something we want to look at. Later on, we definitely would want to repeat this experiment and essentially probably look at the tumor-infiltrated lymphocytes and see potentially if there is some sort of difference in their phenotypes. But so far, we don't know the answer to that.
So, you know whether here the effect of IL-12, right, is on the like a CAR T cell itself or on the other immune cell population?
And we also don't know that just yet. So theoretically, we would say that it's affecting both the host immune cells as well as the CAR T cells, but I can't give you a definitive answer until we probably harvest the brains of these mice to see, one, if the IL-12 is affecting everything and then if maybe the CAR is already still even there in the brain, because they potentially could have either died off, sequestered, or
How could you test that?
that's a good question. I wouldn't know the best way to test that.
You could do it in mice that were deficient in IL-12 receptor, and either the recipient or where you got your cells from. So then they wouldn't
Right.
respond to IL-12.
Okay. I'll keep that in mind. Thank you. Yep. Didn't think about that. Okay. So as I mentioned before, the FLT3 itself potentially looks like it could work, but our CCL3 has shown that it is not working, and then the other candidates are still kind of in testing. So where do we really go from here? And how does IL-12 delivered locoregionally through the CAR T cell impact the tumor microenvironment? All these kind of questions that we've been trying to ask to see, where do we go for the next step? How do we answer these questions? And what experiments do we need to do first to get to these points? So essentially for the first point, is it a role of interferon gamma? Is it a role of TNF alpha? Or is it all kind of come together through T cell activation? Is there any leak or trafficking of CAR T cells out of the brain into lymphoid organs or blood? Some of the things we've been talking about is essentially taking blood from these animals, seeing where these CARs end up being, or if there is any sort of sense of these cytokines or chemokines in the rest of the host organism. Parabiosis is one of the models that we've essentially looked at. I know, Dr. Everett, you don't really like that one, neither do I. But it's pretty scary, but essentially taking a mouse, giving them a tumor, and then sewing it to another one to connect their vascular systems, and then giving that first mouse CAR T cells to treat that tumor, and then giving the other mouse a tumor as well, and seeing if we can traffic those CAR T cells from one mouse to the other to treat. So kind of a Frankenstein experiment, but it's something that a couple of groups, Fetching Group is currently doing, not in relation to CARs. And then what myeloid population is being influenced in these changes? Was it looking at dendritic cell populations, macrophages, T cell activation? There's a lot of questions that we were asking, and we don't really know exactly which foot to put forward just yet, but phenotyping the myeloid populations would be definitely the start from what we would do here, from looking at the brain and blood, and then potentially doing TCR sequencing. Yes, Penny?
So it's like looking at the trafficking of T cells. Could you just like have a luciferase expressing?
Yeah, that is one of the things we've been looking at doing, and then the other thing was potentially treating. What was the other model?
Have you seen anything interesting yet with the luciferase phosphatase?
No, so we haven't done anything with our CARs in that sense, but mainly we've had GFP tumor lines showing that in different scenarios with and without full body irradiation, how the tumor reacts.
Oh, I was
in
saying
terms
like
of-
how the T cell could travel.
Yeah, we don't know that just yet. We haven't done that before, so we'd have to. I think it would be pretty easy to develop, but that is something that we have considered seeing, because that would be a lot easier than doing parabiosis.
Yeah, because like we have like luciferase phosphatase.
Right, yeah.
And it's hard as T cells and then transduce them.
Yeah.
Yeah.
So that could be something that we could do in the near
You could
future.
actually potentially monitor their migration while the mice are alive using one of the in vitro imaging systems.
Yeah, so that's what we did for looking at essentially tumor burden in these mice with our luciferase tumor.
But you could look at CAR burden, to see where they are and where they're going.
Yeah. Right.
So why are you interested in leak?
So this is in terms, I guess, looking at it more clinically. So clinically, we've seen a lot of issues with IL-12 systemically being toxic, and other ideas such as cytokine storms, essentially killing patients and even animals because of this overwhelming burden of cytokine, chemokine, whatever you name it. So looking at that, we want to see if our CARs are still activated and still doing what they need to be doing, where are they doing it, and is that affecting the mouse itself, that burden? So we'd be interested in seeing the kinetics of that.
So is there a difference in the replication rate of these armored CAR T cells versus regular
Yes,
CAR T cells?
the replication is definitely affected when we split these cells. We see generally, I would say there's probably an 18-hour doubling for these cells. And depending on the cytokine or the chemokine, it either gets upregulated significantly or downregulated. So we see that our IL-12s are actually generally more, I'll get this right, I think they're actually, those are upregulated. Our CCL3s are downregulated significantly, and then the rest are more or less on the same baseline as our standard T cells.
So that might have something to do with which ones work, too.
Exactly, yeah, exactly. So my near future aims are essentially, do armored IL-12 CAR T cells result in the induction of endogenous immunity and rejection of a heterogeneous tumor? For that, we'd like to repeat that heterogeneous tumor experiment that Carter did in the previous slides I showed you, because it is the easiest experiment that we can do and just show whether or not this can accomplish the goal that we're aiming for. 2, do CAR T cells engineered to express IL-12 result in enhanced dendritic cell recruitment and maturation? One of the ways we've been looking at that is using a BATF3 knockout to really eliminate our dendritic cell population, and then reinfusing dendritic cells to see how well they'll travel and traffic in terms of depending on the CAR T cell that we're using. And then will a combinatorial CAR T cell therapy elicit host immunity and reject heterogeneous tumors? Here, because we have so many candidates that kind of do different things, we want to look for any sort of synergy that they can play off of. So potentially injecting a 50 dose of IL-12 CARs with a FLT3 ligand CAR, showing because their effects kind of play off of each other, potentially producing the effect that maybe we want to see or what we want to see with just a single CAR alone. And with that, I'd like to acknowledge my lab, Matt Sampson, my mentor, Luis Sanchez Perez, who's also a big mentor of mine, and then the rest of the lab, Carter, for getting me this project, essentially, allowing me to use it, Adam for all his help, everyone else, Tylo, Kelly, Andrew, my medical student, David Snyder, Fetchy Lab, my thesis committee, Soman, Jing Li, Smita Nair, Luis Sanchez Perez, and the pathology department, Meg, doing an astounding job at putting together all this stuff, DLR staff, and all my mice, because I go through so many mice. So, thank you so much, and I'll take some questions.
Just a general question.
Yeah.
Do you know if the absolute size of a solid tumor affects the efficacy of CAR T cells?
No, we haven't looked at that,
So
so.
you're injecting 50,000 cells. If you injected twice, 3 times, 4 times as many, maybe, or even more, do you think it would?
So one of the issues with that is that when we first started using this tumor model, we did a titration to see what tumors will kill, what tumors, at what volume a tumor will take, and develop some sort of curve that allows us to do our treatment and to be able to monitor these mice. And in certain tumor lines, we definitely do have a larger volume injection or smaller volume injection. CD2A parental, generally we do that. It's about a 30,000 tumor volume concentration. So previously Carter did titration with the V3, and with more tumor, there wasn't enough time to actually get those CARs in to actually do the job. They just died off. So we haven't looked specifically at, I guess, how much that tumor volume size is. We always could prepare our T cells in advance to infuse, but no, we haven't seen any correlation between size and burden.
So I'm suspecting that the EGFR3 antigen that these cells are expressing is a human one.
Yes.
So basically, it may or may not be something that's affecting the tumor at all. It's just acting as an antigen.
Yeah. Mm-hmm. So in this sense, kind of using our CAR T cell more as like almost a vessel and
Right.
a targeting vessel.
But the point is it might actually work better in a human cell where EGFR3 actually gives the cell a selective advantage.
That's true.
So,
That's right,
just
yeah.
something to think about, that you're actually doing these experiments under the worst case scenario.
Yeah, definitely the model itself isn't what it really is made to be, but for what it's worth, it's working. Yes.
So most normal T cells, if their antigen's not around, they disappear.
Mhm.
So when you put in your CAR T cells that recognize the EGF3
Mhm.
and it kills them, I
Mhm.
imagine that killing is a lot faster than the endogenous immunity would kill the rest of the tumor. So
Yes.
do you see that, or do you envision that the CAR T cell would kill all of the EGFR3 expressing tumor cells and then kind of die off, and then you sort of lose this efficacy towards the other?
Yeah, so that's kind of what we're expecting is happening, is that the CARs go in, kill off the tumor, those mice live, and then nothing else is going on because the only tumor available there is our EGFRB3 positive. But when we look at a like a heterogeneous, like a 50-50, we see that not working because we believe one of the things is that the CAR leaves, or it isn't able to kill that tumor, and there's no antigen pickup for the rest of the T cells, the host T cells, to kind of do their job. So I guess I don't really know the answer to how to fix that, but like you said, you're correct that it would take a lot longer for the host to kind of be able to be primed to be able to take on the remaining tumor or
And you
heterogeneous.
could be selecting for a CAR-resistant tumor then, I guess.
In that case, you could potentially, yes, yeah. So I don't know the answer to that, but
But the armored CAR T cells are kind of trying to address that.
in a sense, yeah, by promoting this an increased amount of antigen presentation, depending on what type of CAR we're looking at. So potentially being able to take apart these tumors, present more antigen or different antigen to the rest of the host, and then bring about this host killing, rather than just having our targeted V3 killing. Yeah.
Do they need signaling to stick around, the CAR Ts?
From what we've shown, yes, they do need that signaling to stay around. But we treat with so we do also have a wild type EGFR CAR T cell, and we implant those into our V3 positives. They also don't survive.
So the effect the engineer IL-12 in the CAR T cell, right, is really striking.
Yes.
And my question is, do you know what's the mechanism regulate like endogenous IL-12 in the T cell? Whether you can find a way like you can boost the endogenous IL-12 in the T cell?
Yeah, we haven't really looked at that specifically, at how to boost the host to produce IL-12 rather than kind of having our, our kind of delivering this ex vivo CAR in to, supplement with the IL-12. So I don't really know exactly what would be the best way of looking at an IL-12 boost or enhancing the host to do that job for itself rather than using the CAR. But that is definitely something we could look at. Yeah.
And then what effect do you think being in the brain microenvironment has on the success of your CAR cell or your armored CAR cells? And are you planning any experiments to look at the same tumors implanted peripherally to see if there's a difference?
Mm-hmm. So we haven't looked at, so let me address your second question first. We haven't looked at that yet in terms of tumors implanted peripherally, but what we want to do is actually deliver the same armored CARs systemically to treat the intracranial brain tumor to see, one, if they can make it to the brain, and two, does that immense amount of cytokine or chemokine production affect the mouse survival itself. So that's kind of one of the ways we've been trying to address that problem. Can you repeat the first question?
The first problem is whether or not this, we talk about the brain as being a different microenvironment than in other places, and
Yeah.
one where maybe less conducive for immune reactions to go on. And so I wondered
Right.
that if it's the specific microenvironment of the brain is affecting your ability to kill tumor.
Yeah, I would believe so. So in terms of IL-12, we are definitely seeing an influx of interferon gamma. What I was planning to do is essentially take the brains of these mice and see if we are, at least not these current ones I've shown, but in a future experiment after treatment to see if we do see an upregulation of interferon gamma, TNF alpha, to see whether or not that immune microenvironment is being altered in a way that is different from not a naive mouse, but an untreated mouse. But that is something that we still need to look at. That's definitely one of the things we would like an answer to. Also, going back to your question about the host T cells, another group, I don't want to say if it was Rosenberg or not, but they actually transfected just regular T cells, activated T cells, CD8s, that also secreted IL-12, And those T cells alone, without any sort of specific targeting like a CAR, if you remove TBI or total body radiation, those mice also die. So that targeting definitely seems incredibly important in terms of getting those infused T cells to persist and kill. Should I do a student question or?
Just one.
Just one. Zoe?
Most of the research is based on the mice, and what
Environmental factors?
like
Um,
there's
Um,
stress.
Yeah, in that case, some of our groups have definitely looked at, higher elevated levels of cortisol, stuff like that in our mice, or potentially time of day, seeing if that affects tumor growth or how effective certain treatments are. That's mainly been done on the veggie side of our whole unit, but we, at least on our end, haven't seen anything related to any sort of environmental factors. So I don't have a good answer for that. We haven't seen anything related environmentally. Byron.
So these tests, do you, besides looking at mortality, determination, treatment, you also look at like I guess behavioral responses in mice today. So I assume that as tumor grows, there's going to be some change in their behavior. I want to see, of course,
Yeah.
treatment takes place. Does the behavior of the mice return back to normal, or do they end up in like this third like condition where they're not quite back to normal?
Yeah. So based on the timing of our CAR infusions, all the mice are kind of more or less the same. They do have a fully developed tumor, but it's not to the point where I think they're developing very visible neurological issues that we can really monitor, or behavioral issues. Later on, after these mice become more moribund in failed treatments, we definitely see that exhibited probably closer to day 21 through 30. But at that point, I guess we don't really know how to compare that to a control to see whether or not we can really bring them back essentially at that point. So yeah.
So from previous experience, it seems like CAR T cells may have some adverse effects on patients, like maybe cytokines, the one for
Yeah.
like pro-inflammatory syndromes. So do you think that there's some studies like studying how we can maybe turn off the CAR T cells by some kind of maybe tamoxifen use?
Yeah, there definitely are those. So, like I said, in most of these armored cards that I've made, these are currently all constitutively secreting. We have not made them, but we have the ability to make an inducible card with, like you said, either tamoxifen or doxycycline. That's what we've been looking at in terms of turning off the gene that secretes our cytokine. There are also cards that have an on-off switch. I can't remember the exact name. They have some fun name like convertible or something like that too. But there are cards that do that. Yes.
And there are some that have suicide genes, so that
Exactly,
if
yeah.
they have a cytokine storm, they can suicide.
Exactly.
So
Yeah.
wouldn't be good to give your patients tamoxifen that's a carcinogen.
Yeah. So I mean, in our case,
It's an anti-carcinogen for breast cancer.
yeah. But in
Depending on cancer.
Yeah.
yeah, in this model, essentially, yeah, it's looking at doxycycline, and we haven't developed that yet, but we want to look at. Constituent was the easiest one to get out to first look at to see if there is some sort of mortality.
I'm just wondering in the clinic, how many T cells did they get per patient within the license? 2 million, you said 2 to 6?
Yeah, 2 to 6. I can't give you an exact number. It's a lot. It is a lot of IV bags full. Actually, Kelly, would you have a better answer because you've been actually doing on the clinical side?
I know we only got like 30% of what we wanted to inject it, but I don't remember what the number was
Yeah,
before it clotted. But
I think most of the time they do inject, they plan for more because how they're delivered, at least intracranially, these cells can not necessarily clot, but they're almost like atherosclerotic, but they'll stick to the side of the tube, and you don't get a full delivery or a full dose, and it's also hard to tell how much you're giving them because of that. So there's still a lot of troubleshooting.
And they're from the same patient, right?
Yes, yeah. So it's hard to tell, but it is a lot more in terms, even ratio-wise, compared to mice. Yeah. Awesome. Thank you, guys.
All right, thank you. So when I did something that happened. So I was talking to the students after. They were all like, oh, I don't know.
Yeah, is that the only people we have in John's lab?
I don't want to be academics. Well, I was, when you future, and then I was like, why? This is very hard for people to survive.
I looked at my.
Then he said also, and we are all of them saying the same thing. We're from, and then I was, I was more.
Great job, Steve.
So you should know.